Welcome, everyone. Thank you for joining today's webinar on exploring integrative structures with RCSB.org. I'm Brinda Walad. I'm a member of the RCSB Protein Data Bank at Rutgers University, and I'm a scientific software developer, and I work with different data-related projects here at RCSB PDB. Over the past few years, I've been working on a project focused on archiving macromolecular structures determined using integrative and hybrid methods in the PDB archive. So today, I'm going to provide an overview of what integrative structures are, how they are archived in the PDB, and what resources are available on rcsb.org to explore and analyze these structures. Let me begin with an introduction to the Protein Data Bank. As you all may already know, PDB is the single global repository for experimentally determined structures of biological macromolecules and their complexes. It was established in 1971 with 7 structures. It currently contains over 250,000 or approximately 250,000 structures of macromolecules. PDB is the first open access digital resource in all of biological sciences. And the PDB promotes the FAIR guiding principles, which makes structural biology data findable, accessible, interoperable, and reusable. The PDB archive is managed by the Worldwide Protein Data Bank organization, consisting of the RCSB PDB here in the United States, Protein Data Bank in Europe, Protein Data Bank Japan, the Biological Magnetic Resonance Data Bank, or BMRB, and the Electron Microscopy Data Bank, or EMDB. The PDB archives structures of macromolecules determined using X-ray crystallography, NMR spectroscopy, and 3-dimensional electron microscopy. The structures archived in the PDB contain 3D atomic coordinates and associated metadata. The PDB also archives structures that are determined using multiple methods. These methods usually use one of the traditional structure determination methods, such as X-ray, NMR, or 3D EM in combination with other methods, such as neutron diffraction or small angle scattering. In recent years, structures of many macromolecular assemblies have been determined using integrative modeling methods. So what is integrative structure determination? It is an innovative approach to modeling the structures of biological systems based on data produced by multiple experimental and theoretical methods. It is typically used for modeling the structures of complex macromolecular assemblies that are not amenable to traditional methods of structure determination or single methods of structure determination, such as X-ray crystallography, NMR spectroscopy, or electron microscopy. Integrative modeling involves combining data from multiple experimental methods, such as, and it includes X-ray, NMR, EM, and other methods like small angle scattering, chemical cross-linking, Foster resonance, Fred spectroscopy, and Foster resonance energy transfer, Fred spectroscopy, electron paramagnetic resonance, hydrogen deuterium exchange. These are other biophysical methods and proteomics methods that contribute data to integrative modeling. The experimental data from these different sources are gathered and converted to spatial restraints and combined with known starting structures of molecular components that may be available. And these are combined together using computational modeling algorithms to build structures of complex macromolecular assemblies. So this is typically what we consider as integrative structure determination. So then the question is, can the PDB archive integrative structures? Initially, there were some technical challenges to archiving these structures in the PDB archive, and these challenges arise due to various factors, including that integrative modeling involves restraints from these different experiments like cross-linking or FRET or small angle scattering, which are not supported by the core PDB archive. And integrative models can be multi-scale or multi-stage, and I will come back to this and explain these concepts in detail. But for now, these are factors that kind of affect how these structures are archived. These models can also be conformational ensembles and models that have heterogeneous composition. And heterogeneous composition is also something that is not supported in the core archive. And integrative models use starting models coming from experimental and computational methods, and they use different modeling methodologies and software applications. So these factors affect how these models are archived and require some special considerations. So in order to address these challenges, the Worldwide Protein Data Bank set up a task force consisting of researchers from different fields contributing to integrative structural biology. The first meeting of the task force took place in October 2014 at EBI. And this task force came up with a set of recommendations to archive integrative structures. I've summarized it here, but I don't want to go into the details. Just want to highlight a couple of bullet points that are important for this presentation, this webinar. One important aspect is that the task force recommended adopting a flexible model representation, which again corresponds to the multi-scale, multi-state assemblies, and I'll explain that in the next few slides. And also, they recommended that methods be developed for assessing the uncertainty of integrative structure models. So these are important factors that I will touch upon in the rest of this webinar. So at the PDB, we started with the goal of building a system for archiving integrative structures based on these recommendations from the community. So what are the requirements for archiving integrative structures? The first requirement is the creation of data standards. Data standards are the primary requirement for archiving and enable automated data processing as well as dissemination of data in a standard form. And from PDB perspective, data standards typically encompass definitions for the chemistry of macromolecules and small molecules, how we represent polymeric macromolecules and small molecules in the structures archived in the PDB, and the 3D atomic coordinates, as well as any other information, metadata, or information about these structures that we want to capture. So that's what needs to be encompassed in what we call the data standards. And then the next requirement is to create tools and the infrastructure, the pipeline needed for data deposition, processing, archiving, and dissemination. This is what we call the data pipeline. So let's look at the first requirement, which is the data standard. Luckily for us, we have a strong foundation to begin with, which is the PDB-IHM data standard used by the PDB to archive experimental structures. In case you're not aware of PDB-IHM, I encourage you to visit the MMSIF website. The link is provided here to learn more about it. PDB-IHM specifies the standards for representing macromolecular structures, including the chemistry of polymeric macromolecules and small molecules, atomic model coordinates, additional metadata about source organisms, samples, authors, software, etc., and also reference information about these molecules, whether it is a polymeric macromolecule or a small molecule. Additional reference information is also included in these definitions. And PDB-IHM is extensible and has software support to read, write, and maintain these the files that we have in the PDB archive. And you can learn more about it on the MMSIF website. So we decided to extend the PDB-IHM MMSIF data standard for integrative modeling, and this extension is called IHM SIF. And IHM here stands for Integrative and Hybrid Methods. We retain the standard definitions for representing macromolecules and small molecules from PDB-IHM MMSIF, and the new definitions that are required for integrative modeling are defined in IHM SIF. And this includes definitions for multi-scale, multi-state, ordered state and ensembles, compositionally heterogeneous assemblies, the different kinds of experimental restraints, starting models, and workflows that are used in integrative modeling investigations, and also relevant information about experimental data that may be available in other resources. We also have software tools that support the IHM SIF dictionary, which can be used to programmatically generate files that are compliant with the dictionary. It's called the Python IHM toolkit. Both IHM SIF and Python IHM are freely available on GitHub. So now I want to explain the different components of the dictionary and primarily what we mean by multi-scale, multi-state, ordered ensembles. Ensembles are sets of models that correspond to multiple solutions that individually satisfy the input restraints within a certain threshold. So they're all independent solutions, but they satisfy all your input restraints within a certain threshold. Multi-scale refers to models that have both atomic and coarse-grained representations, and the coarse graining can be at different levels. You can coarse grain as a single residue bead or multi-residue beads, or even at a domain level. So that is what we refer to as multi-scale. Multi-state models correspond to models that are in multiple conformational or compositional states that collectively satisfy the input information. That's how they are different from the ensemble representation, where each one individually satisfies the input representation, whereas in multi-state, you can have, say, an open or a closed conformation, or a bound or an unbound conformation, and they collectively satisfy the input restraint, which can be very heterogeneous. And you can have ordered state, which is a higher level representation of multi-states. These are multi-state models that can belong to either a time-ordered path or another ordered path, such as an enzymatic reaction or biochemical pathway. And you can represent the system as an ordered state that satisfies the input restraints. In addition, we can have any combination of these features, these four features, in an integrative model. For instance, you can have a multi-state model that has both atomic and coarse-grained representations. So these are not mutually exclusive. So as recommended by the task force, we've adopted a flexible model representation that can handle multi-scale, multi-state ordered ensembles. The reason this is important is this is how we represent integrative structures in the PDB archive, and knowing this will help you explore these structures on rcsb.org. Another important factor about integrative structures is the diverse kinds of spatial restraints and starting models that can be used in integrative modeling. Experimental and computational models, by computational models, I mean it could either be homology models or ab initio models. They can be used as starting models in integrative modeling. At the same time, you can have a variety of spatial restraints derived from different kinds of experiments. Cross-linking experiments give you distance restraints. You can have 2D EM class averages, SAS profiles, 3D EM maps that can be different 3D EM maps representing different parts of an assembly that is being modeled. You can bring all that together to carry out an integrative modeling investigation. And this information is represented in the IHM CIF dictionary and captured in the PDB-IHM archive. And we also have, to maintain provenance, we also capture the source of where these input information is coming from. If you have structures, models from AlphaFold, include that information. And then if you have data from cross-linking that is in a resource like PRIDE at EBI, or SAS from SAS PDB, we include that source information so that we can have provenance, and we can also use that for validating the structure models. Additionally, we also capture generically the modeling workflow. The integrative modeling workflow also can be very, very diverse, but in general, you can define it as you gather all the information, represent your system that's being modeled, and then you have some kind of sampling and scoring and post-modeling analysis. So those are generically the different steps involved. And we have ways to capture that information. So, and why it's important to capture this information is knowing how the modeling was done makes it easier to interpret the structure for any downstream applications. So with all these different components, the integrative model itself, the starting models used, the experimental restraints, and the workflow, with all those definitions and with all those components, we have been able to build a system for archiving integrative structures. And this is a representation of the data pipeline where modelers or depositors can come in and deposit structures, including the coordinates, spatial restraints, starting models, etc. And that gets taken up by our data processing pipeline, where we carry out curation, validation, visualization. And then the structures are archived and distributed. And I will focus on the data distribution aspect so that you're able to explore these structures on rcsb.org, but I will quickly cover the rest of it so you have a background information of how this is being done. With regard to deposition, the deposition itself requires IHM CIF compliant files, which have files that have complete information about the model coordinates, the molecular system, the restraints, and reference information and other metadata. But we also understand that it can be a bit too much to provide all that information. So there are multiple ways to do that. You could use the Python IHM library to create compliant files prior to deposition and then breeze through the workflow. Or we have a data harvesting section of the deposition pipeline, which is accessible from this URL. And you can start with a minimalistic structure, just the coordinates, and add all other information progressively. But that would be time-consuming, of course. But there are multiple ways to address the requirements. And we also have an API. If you have a compliant file, you can use the API to submit multiple structures. With regard to biocuration, it is similar to what is done in the core PDB biocuration pipeline. We check for data consistency and compliance with the dictionaries, make sure that the nomenclature is consistent with what is used in the core PDB, and then issue accession codes and generate validation reports. With regard to visualizing integrative structures, there are at present two software applications that support. You have the desktop Chimera X software that supports visualizing integrative structures, as well as the Molestar web application. In my demo today, I will go through the Molestar web application that we use on rcsb.org. But if you're interested in using Chimera X, I just want to point out a question that usually comes up is, oh, I took a structure from PDB-IHM and it doesn't work. So Chimera X doesn't natively support IHM CIF. You just need to tell it that this is an IHM CIF file. This is a PDB integrative structure, and use a command line argument that I have put here. You could use that to visualize structures using Chimera X. And the next part of our pipeline is validating integrative structures. And this is, again, important because going back to the community recommendations, one of the recommendations was to develop methods for assessing model uncertainty for validating integrative structures. And based on these recommendations, we've collaborated with the community to develop methods for validating integrative structures. There are different assessment categories. Again, these are based on recommendations from the community that evaluate the data quality, the model quality, and how the model fits the data that was used in the modeling. And currently, we support validation for 3 different input data types: small angle scattering, cross-linking, mass spec, and 3D EM. And this is because primarily most of the integrative structures fall into one of these or use one of these input data types. So we prioritize that, but we continue to develop other methods as well. The validation software is integrated into our deposition and curation pipeline. These validation reports can be obtained after deposition and can be used for manuscript review process. Summary and full validation reports are available. And recently, we also deployed a standalone validation server where before deposition, you could upload your structure and get a validation report. But again, the requirement is that you should have a near complete file so that the validation report is meaningful. If you don't have your input restraints, then the validation is not going to give you that information. So it's important to have a near complete file so you get meaningful validation information. So I wanted to highlight some of the important aspects of what we do with validation. Again, this is based on feedback from the task force and from the community. We support small angle scattering, cross-linking MS, and 3D EM-based structures, validation of these structures. 3D EM validation is based on what is currently implemented and followed by the wwPDB validation reports. And for SAS and cross-linking, we collaborated with the respective communities to get their recommendations and incorporated that into the validation pipeline. And we created methods to methods for data exchange or to interoperate with these other resources like EMDB, SAS BDB, and PRIDE, because the data that was used for modeling is available in these resources. So we want to be able to use that for validating these structures. And so we created mechanisms to interoperate with these resources. Specifically with PRIDE, which is a proteomics repository, we work with them to create a new section of PRIDE called PRIDE Cross-Linking, which now supports submission of complete cross-linking MS datasets. And we encourage depositors to use that resource to submit complete cross-linking data to PRIDE. What I'm showing here are some of the snapshots from the validation report for cross-linking EM and SAS. And I wanted to highlight a particular use case here, which is, I think this is the structure of Fanconi anemia core complex, which uses 3 different EM maps from EMDB that corresponds to different parts of the assembly. So the EM maps are used to model the whole complex. And then the cross-linking data, cross-linking restraints are the inter-subunit crosslinks help bring these maps together to build the entire complex. So this is a typical use case for integrative modeling, and I just wanted to highlight how the validation report can tell you, okay, there are 3 different maps and these crosslinks, and what's violated, what is satisfied. You can get that kind of information from the validation reports. Okay, so moving on, we created the pipeline for archiving integrative structures, for representing the complex data standards, for deposition, curation, validation, visualization. All these was created in parallel. And we developed it separately from the core PDB to kind of enable an agile development of this infrastructure required for archiving integrative structures. However, from the very beginning, our goal was to create the infrastructure and then unify it with the PDB so that we can expand the capabilities of the PDB to archive integrative structures. And in the past year, we did that. PDB-IHM is now unified with the PDB archive, and subsequently, it's also now available on rcsb.org. This figure shows the current PDB-IHM data pipeline, including the newly added extensions that support integration with the PDB archive. The PDB-IHM deposition system can now be accessed from OneDep. The deposition system itself is same, nothing has changed. You need to submit the integrative structures and spatial restraints and additional metadata, but you can access it from OneDep. And same thing with the data processing. The processing is the same, hasn't changed. We have the bi-curation, visualization, validation pipeline, but now we provide PDB accession codes and digital object identifiers for integrative structures. So that's the new extension. And we created workflows to support synchronized weekly update process. So the release of integrative structures is synchronized with the release of experimental structures from the wwPDB OneDep system, so that the structures are released at 0 UTC on Wednesdays, following the same practice as core PDB. And if you look at the PDB archive, there is a parallel directory. Parallel to PDB, there is a PDB-IHM directory that now contains all the integrative structures that are processed by the PDB-IHM pipeline. And this, the PDB archive along with PDB-IHM is mirrored by all the wwPDB partners. And this now enables RCSB.org to consume that data along with experimental structures and then deliver it on RCSB.org. So going to the most important part of the talk, which is how you can explore these structures on rcsb.org. RCSB website supports search, access, analysis, and visualization of integrative structures, similar to what you would find for experimental structures in the PDB. Just like you can search and analyze and visualize experimental structures, we have tools to do the same for integrative structures. And this integration was made possible primarily because the IHM CIF data standard is an extension of PDB-XMM MSIF, which is used by the PDB archive. Before I go into the demo, I just wanted to highlight or give you an overview of what's going to come. We have all our APIs, all RCSB.org APIs and search services now support integrative structures. Individual structure summary pages are available for these structures, and they can be visualized using Molestar on the website. Similarly, all the added annotations that you can find from external resources for experimental structures, they're also available for integrative structures. In addition, IHM specific information like multi-scale, multi-state structures, or experimental data like crosslinks and SAS and other information from other resources, how to find relevant information from other resources, all that is additionally incorporated for integrative structures. I also want to point out that all this primary and computed data and all these external annotations are processed and loaded for a representative model. And this is consistent with what is done for experimental structures in the PDB. So if an entry contains multiple structure models, for example, an ensemble, then during deposition, depositors can select one of the models to be the representative confirmation. And if this is identified and provided by depositors, then the representative model is identified and the annotations are loaded and information is loaded for that representative model. But if it is not provided by the depositor, then we choose the first model containing the largest number of molecules as the representative model and load information for that model. Okay, now I want to switch my screen and go to I want to share my all right. Okay, let's move to the demo part, how you can explore integrative structures on rcsb.org, the tools and functionalities that we have on rcsb.org. So this is our homepage. We have, as you can see here right on top, we have structures from the PDB archive and computed structure models that we've incorporated from AlphaFold and Model Archive. They are also available, but the core PDB itself now consists of both experimental structures and integrative structures. We can identify the experimental structures that are identified using the conical flask icon, and integrative structures are identified using a puzzle piece icon. And this is a theme that you will see all through. So I just wanted to highlight that. And before we move on, there are some other resources, documentation, which are available. If you click, you can find this. This kind of covers what the introduction, the background information that I just presented. What are multi-scale models, why integrative structures matter. You can find this information on our website. And let's now click on this puzzle piece icon, which will directly take us to the set of integrative structures, which currently is 381 in the PDB-IHM branch of the PDB archive. So just clicking that will directly take you to the list of integrative structures. If you look at what was launched when you clicked that icon, there is a specific term called structure determination methodology that is set to integrative. And that's how that is specifically identified in the MMSIF file. It is available in the archive, and that's to consistently identify and distinguish integrative structures from experimental structures. That's the attribute that can be used to get that information. Now, scrolling down at the search results, the structures are identified using the puzzle icon. There is the structure determination methodology that tells you these are integrative structures, and then the rest of it is very similar to what you would find for experimental structures. The release date and title, authors, that kind of information. Then I want to draw your attention to the refinement panel here that helps you drill down further into your results. If you've used rcsb.org before, you will know that these are generic. These things you will find for experimental structures as well: organism name, taxonomy, and all that. But I want to bring your attention to integrative input data, which is, again, specific for integrative structures. And this helps you drill down based on what kind of experimental data was used in integrative modeling. For example, if you want to use cross-linking MS, you can click that and refine, and it'll give you the subset of structures that are based on cross-linking restraints. All right. Next, let's start with an example on the search bar here. And let's say we do BB-SOM, which is a complex. We launch a search, and that will bring us to the search results page where you have both experimental and integrative structures that satisfy that search criteria, which is BB-SOM. And then you can, again, use the refinement panel if you want to drill down and see if you just want the experimental structures or integrative structures. You could do that. I want to next highlight how you could use the advanced search menu to explore integrative structures. And as I mentioned before, we have specific attributes that are relevant to integrative modeling. And if you go to our structure attribute search and type IHM, you get a set of IHM-related attributes that you can explore. Let's say you want to do multi-scale flag. You want to look at all the multi-scale structures. Search that, set that to yes, and then you get a set of results that are multi-scale. Or if you only want atomic, you could turn that off. And you can combine the attribute search with other attributes. For example, I want to demonstrate how you could use search for atomic structures that are based on, let's say, cross-linking MS data. And then you can combine that to search for these structures. In addition, maybe you want to also look at cases where you have AlphaFold models were used as starting models. So then you can say the source database from AlphaFold is not empty, which means you have an accession code from AlphaFold. That you have a cross-linking structures based on cross-linking MS data and is atomic. So you can have create a complex query and search for that, and you get 7 structures that satisfy that criterion. So these are all IHM specific. You don't have to restrict to that. You can combine it with other attributes that are relevant for all structures. Let's say you want to see deposited model count, which is true for, experimental or integrative. And then you can say, I want it to be greater than or equal to 10. I want cases where there are multiple models. Maybe you want to look at conformational changes, or you want to see how multi-state modeling is done. Sorry. So you can incorporate other attributes here as well. Okay. So we looked at the full text search, attribute search, and the power of RCSB.org search is fully available when you're able to combine these kind of searches with other scientific searches. So let's say you start with these attributes, and then you want to also add a sequence similarity, which is a scientific search. Let me grab a sequence from my notes. Oh. Okay, so you can add a sequence similarity to this complex query, and okay, when I add sequence similarity, I need to change my return to polymer entities because at this point, I'm specifically looking for polymeric molecules. And then I can get specific results that that are based on these attributes, as well as combining it with the scientific sequence similarity search. All right. Okay. So then once you've been able to search and get some results, let's say you want to go explore the structures, and click on one example here, and you go to the structure summary page. It's very similar to what you will see for experimental structures. You have the accession code information, deposition date, and authors, and all the generic information. But I want to draw your attention to the snippet here that says integrative models are generated using different types of input information, and that shows this is an integrative structure. And you also have an integrative structure snapshot that tells you whether it is a multi-scale, multi-state, and how many deposited models, and what is the representative model. That's very useful to know. And you have access to full and summary validation reports. Just clicking on it will give you the PDFs. And you can also go to the download button here, download files, and you can get the validation reports and all the MMSIF files and for download. Scrolling down, you could get all the macromolecular information similar to what you will find for experimental structures. And if you have small molecules, I think I have an example here, maybe. Yes. This one, for example, has more deposited models, and identifying the representative model is important. So you can see what happens when you have multiple models. And if you have small molecules in the structure, then you'll be able to see that as well, similar to experimental structures. Next, I want to move on to the structure tab. This is all about the structure summary page. If you go to the structure tab, it launches our MolStar viewer in atomic structures, very similar to what you will see in experimental structures. You can visualize the structure. But I also wanted to point out a use case where it is not atomic. You go to the structure tab, and you can see that this is a multi-scale structure that has beads of different sizes, coarse-grained beads. And you can, MolStar is able to visualize these structures. This example also happens to be an example that uses crosslinks. So I want to show another functionality here where you can visualize crosslinks. MolStar supports that. So if you go to the course representation and add representation, here you could see that there is a crosslink representation, and clicking on that will show you the different crosslink restraints that were used in the modeling. And if you hover on the crosslink itself, you can see in the bottom there, you can see the information about the crosslink. It's a upper bound restraint. The threshold was 16 angstrom, and the model distance is 25, which means the crosslink is not satisfied, it's violated. In addition, you can also, once you have the crosslink representation, you can also change the color theme to show satisfied and violated crosslinks. So the coloring now is based on satisfied and violated crosslinks, and this tooltip here shows you what the color coding is. The crosslinks that are deviated within this range are color coded based on what's shown here. So the black ones are, of course, violated by 1.5 or more angstroms. So that's a feature in MolStar to visualize crosslink restraints. We are working on doing the same for other kinds of restraints, but crosslink, since it's very frequently used in integrative modeling, this is an important feature. All right, so going back to our example, after the structure, we go to the annotation tab, and RCSB.org incorporates annotations from 50 odd external resources like Gene Ontology and Interpro. And this is done for, in addition to experimental structures, the same thing is done for integrative structures. And you can find all these annotations, external annotations, in the annotation tab. And importantly, if you go to the experiment tab, you can find information about the different kinds of experimental data that was used in the integrative structure modeling. In this case, you have 3D EM maps, cross-linking data, starting models from experimental and ab initio methods, and also links to data from other resources. So the cross-linking data from PRIDE, the maps from EMDB, and the starting models from AlphaFold and PDB. So with one click, you can access all that information. Moving on, we have the sequence tab, which provides annotations, maps the annotations to the sequence. If you've explored rcsb.org before, you might have seen this for experimental structures. It's very similar to what you would see in those cases. The annotations are mapped to the sequence. I wanted to go back to and show an example of what we call uniprot groups. So starting from the structure summary page is one way to get to structures. The structure is the main component, and you want all information about the structure in terms of annotations, experimental data, sequence information, etc. Another way to explore rcsb.org is, let's say you start with a sequence itself or a uniprot accession, and you want to know all other structural information that's available for that particular sequence or a uniprot ID. So you're approaching this same problem from a different angle. You're starting with something else. So for such use cases, we have what we call a uniprot group. You can click here, or we could launch. Let me start with, I have the same uniprot. You can search for a uniprot accession on our full text search, and it takes you to the group sequence page where you see the uniprot sequence and together with other sequence annotations and all the different structures that are available for the sequence. And the nice thing about this view, and I personally use this quite a lot, is you can see which structures map to which part of the sequence. You can see there are 3 PDB structures that actually map to different parts of this uniprot sequence. There is the AlphaFold model for the entire sequence, and you can also see how reliable the AlphaFold models are based on the color coding here. And also this integrative structure, which has kind of incorporated everything together with the EM map and the cross-link restraints to model the entire sequence. So that kind of information you can get from this the group sequence view. And if you click on Explore the sequence in 3D, you get the same sequence view, but also you can map it to the 3D structure. Let me just turn this on for these different structures. And as you move through the sequence window, you can see where it maps to the 3D structure here. And you can turn things on and off and specifically analyze if you want to just see how the integrative model maps to the AlphaFold, or if you want to specifically see how it maps to this, how it aligns with this PDB structure, you can do all that, explore that using the 3D view here. All right. Finally, I wanted to, let's see, I want to show another feature, which is pairwise structure alignment. And pairwise structure alignment, currently we support for integrative structures, but only the ones that are atomic. let's say we look at, sorry, where did I have the, okay. Let's say we want to look at 9AAG and 7SFZ. So I can do 9A8G and 7SFZ. I'm just going to pick the default ones here and click compare. And this is an atomic structure, so we can do pairwise alignment. And very easily, you can see how 9AAG, as we saw in this group sequence view, it has the entire sequence, but then 7SFZ is only a part of that sequence is covered by 7SFZ, and you can see where that is and how that aligns. You can get other information about what is the RMSD and TM score, how well the alignment is. You can get some information about that. And you can use the pairwise structural alignment tool, as I mentioned, for atomic structures. Okay, so with that, these are all the different features on rcsb.org that you can use to explore and understand and analyze integrative structures. Let me now go back to my presentation. Sorry, could you just give me 1 second? All right. So we are close to the time, so I will focus just on wrapping up at this point so we can open it up for questions. I have some slides about the current status, but I don't want to focus on that at this point. This is just a snapshot of different kinds of structures that we have in PDB-IHM, but you'll be able to explore it on your own and understand these structures. This slide here shows the statistics for different types of experimental methods used in determining structures. I just wanted to highlight that cross-linking mass spec kind of looms large. So many structures are based on cross-linking mass spec. That is the predominant method, but you also have the combination with 3D-EM and NMR and SAS is also used widely. We did an analysis in 2023 of the primary publications corresponding to low-resolution 3D-EM structures in the PDB, and that showed that about 25% of these structures are likely to be integrative structures. So we want to, and many of these structures are not annotated as integrative structures and using other kinds of restraints, maybe even from crosslinks. So PDB-IHM provides a robust method for archiving these structures and making this data findable, accessible, interoperable, and reusable. And we are also able to do enhanced structure validation if these structures are annotated and deposited through the PDB-IHM mechanism. So what are our next steps? We want to encourage the community to improve deposition practices, encourage deposition of integrative structures to PDB-IHM, especially the 3D-EM structures that use additional experimental restraints. We will continue to support evolving methods. Recently, we had the first deposition of integrative structures based on atomic force microscopy. So as new methods are used, we will continue to support them. And we're also developing improved methods of validation, including other methods. Now we have SAS, EM, and cross-linking. We want to include other methods and as well as develop a comprehensive Bayesian approach for structure validation. And from a big picture, we're also working on creating a federated network of data resources that contribute to integrative structural biology. Like we now have methods for interoperating with PRIDE and SAS-BDB, whatever other resources are contributing to integrative structural biology, we want to be able to interoperate with them. With that, I will move on to some housekeeping announcements quickly. PDB is transitioning to extended PDB IDs. We, believe it or not, we are running out of 4-character PDB accession codes, and these will be fully allocated by 2028. And when that happens, support for the PDB format will not be continued. People will have to transition to PDB-X MMSIF, which will become the primary format going forward. And we have several resources for people to transition. On the wwPDB website, there is a link here. Please go if you want to find out more. If you have workflows that are dependent on the PDB archive, you would want to look at this sooner than later. And PDB-IHM will also transition to the extended PDB IDs. And finally, I would like to thank all my colleagues here at RCSB PDB who have contributed to not just creating this very, very valuable resource, but for this specific project of incorporating IHM, it has been a great team effort. Everyone has contributed, and I would like to thank everyone, our director, Stephen, Director Emerita, Helen, and Andre Shali, who is spearheading the PDB-IHM effort specifically. So, and we are funded by NSF, NIH, and DOE, thanks to our funding agencies. And the PDB-IHM project is a wwPDB project, which means we have worldwide collaboration. So I wanted to thank all our wwPDB directors and task force members, the community who have contributed and who've been supporting this effort over the past several years. And PDB-IHM was also funded by NSF awards. So, and thank you all for your attention. I'm happy to answer any questions.